Now, the processes of recombinant DNA technology. Recombinant DNA technology involves several steps in specific sequence such as isolation of DNA, fragmentation of DNA by restriction endonucleases, isolation of a desired DNA fragment, ligation of the DNA fragment into a vector, transferring the recombinant DNA into the host, culturing the host cells in a medium at large scale, and extraction of the desired product. So let's examine each of these steps in some details. Isolation of the genetic material, that is DNA. So recall that nucleic acid is the genetic material of all organisms without exception. In majority of organisms, this is deoxyribonucleic acid or DNA. In order to cut the DNA with restriction enzymes, it needs to be in pure form, free from other macromolecules. So since the DNA is enclosed within the membranes, we have to break the cell open to release DNA along with uh, other macromolecules such as RNA, proteins, polysaccharides, and also lipids. So this can be achieved by treating the bacterial cell or plant or animal tissue with enzymes such as lysozyme for bacteria, cellulase for plant cells, chitinase for fungus. You know that genes are located on long molecules of DNA intertwined with proteins such as histones. So the RNA can be removed by treatment with ribonuclease, whereas proteins can be removed by treatment with protease. Other molecules can be removed by appropriate treatments, and purified DNA ultimately precipitates out after the addition of chilled ethanol. This can be seen as collection of fine threads in suspension. Now the cutting of DNA at specific locations. Restriction enzyme digestions are performed by incubating the purified DNA molecules with the restriction enzyme at the optimal conditions for that specific enzyme. Agarose gel electrophoresis is employed to check the progression of a restriction enzyme digestion. DNA is a negatively charged molecule, hence it moves towards the positive electrode, means anode. The process is repeated with the vectored DNA also. The joining of DNA involves several processes. After having cut the source DNA as well as the vector DNA with a specific restriction enzyme, the cutout gene of interest from the source DNA and the cut vector with space are mixed and ligase is added. This results in the precipitation of recombinant DNA. Now the amplification of gene of interest using PCR. PCR stands for polymerase chain reaction. In this reaction, multiple copies of the gene or DNA of interest is synthesized in vitro using two sets of primers, small chemically synthesized oligonucleotides that are complementary to the regions of DNA in the enzyme DNA polymerase. The enzyme extends the primers using the nucleotides provided in the reaction and the genomic DNA as template. If the process of replication of DNA is repeated many times, the segment of DNA can be amplified to approximately billion times, that is 1 billion copies are made. Such repeated amplification is achieved by the use of a thermostable DNA polymerase isolated from a bacterium Thermus aquaticus, which remain inactive during the high temperature induced denaturation of double-stranded DNA. The amplified fragment, if desired, can now be used to ligate with a vector for further cloning. Now, insertion of recombinant DNA into the host cell or organism. There are several methods of introducing the ligated DNA into recipient cell. Recipient cells, after making them competent to receive, take up DNA present in its surrounding. So, if a recombinant DNA bearing gene for resistance to an antibiotic, example ampicillin, is transferred into E. coli cells, the host cells become transformed into ampicillin resistant cells. If we spread the transformed cells on agar plate containing ampicillin, only transformants will grow. Untransformed recipients cells will die. Since due to ampicillin resistance gene, one is able to select a transformed cell in the presence of ampicillin. The ampicillin resistance gene in this case is called a selectable marker. Now, obtaining the foreign gene product. When you insert a piece of alien DNA into a cloning vector and transfer it into a bacterial, plant, or animal cell, the alien DNA gets multiplied. In almost all recombinant technologies, the ultimate aim is to produce a desirable protein. Hence, there is a need for recombinant DNA to be expressed. The foreign gene gets expressed under appropriate conditions. The expression of foreign genes in host cells involve understanding many technical details. After having cloned the gene of interest and having optimized the conditions to induce the expression of the target protein, one has to consider producing it on a large scale. Can you think of any reason why there is a need for large scale production? If any protein encoding gene is expressed in a heterologous host, it is called a recombinant protein. The cells harboring cloned genes of interest may be grown on a small scale in the laboratory. The cultures may be used for extracting the desired protein and then purifying it by using different special separation techniques. The cells can also be multiplied in a continuous culture system, wherein the used medium is drained out from one side while fresh medium is added from the other to maintain the cells in their physiologically most active log or exponential phase. This type of culturing medium method produces a large biomass, leading to higher yields of desired protein. Small volume cultures cannot yield appreciable quantities of products. To produce in large quantities, the development of bioreactors, where large volumes like 100 to 100,000 liters of culture can be processed, was required. Thus, bioreactors can be thought of as vessels in which raw materials are biologically converted into specific products, individual enzymes, etc. Using microbial plant, animal, or human cells, a bioreactor provides the optimal conditions for achieving the desired product by providing optimum growth conditions like temperature, pH, substrate, salts, vitamins, oxygen. The most commonly used bioreactors are of stirring type. A stirred tank reactor is usually cylindrical or with a curved base to facilitate the mixing of the reactor contents. The stirrer facilitates even mixing and oxygen availability throughout the bioreactor. Alternately, air can be bubbled through the reactor. If you Look at the figure closely. You will see that the bioreactors has an agitator system, an oxygen delivery system, and a foam control system, a temperature control system, pH control system, and sampling ports so that small volumes of the culture can be withdrawn periodically. Now, downstream processing after completion of the biosynthetic stage, the product has to be subjected through a series of processes because it is ready for marketing as a finished product. The processes include separation and purification, which are collectively referred to as downstream processing. The processed product has to be formulated with suitable preservatives. Such formulation has to undergo thorough clinical trials as in case of drugs. Strict quality control testing for each product is also required. Thus, the downstream processing and quality control testing vary from product to product.